Hello everyone, my name is Ferry. So today I will share my recent study about using DeepLabCut for Markless cardiac physiology and toxicity estimation in water fleas or Daphnia magna. First of all, we have to know what is Daphnia magna and why they are useful for ecotoxicity studies. Daphnia magna is a small planktonic crustacean that lives in water bodies. They can live everywhere in pond, lake, paddies, or even in water puddle. Why they are used for ecotoxicity studies? Because they have a high reproduction capability. One female offspring can lay about 10 eggs, and after 1 or 2 weeks, it already developed into adult and can again lay some eggs. The second reason is because they have transparent body. This transparent body makes us easier to look for their inner organ like their heart, their egg sac, their gastrointestinal trails, and other things inside the body. And the other things is Daphnia also already approved by OECD, which is uh, make them really useful and a valid animal for use in toxicity studies. And with recent development in machine learning, especially in computer vision field, a lot of detector has been developed to track the behavior of animal like single shot detector, UNet, RFCN, YOLO, eigenface, StackNet, or scale invariant feature transform. And the recent one is mass RCNN that's been used to calculate the cardiac parameter in Daphnia magna. However, the last study have some limitation. First of all, it prone to human error because although the detection done by machine, the calculation still done by humans, so it's still prone to human error. The second reason is because of overestimation, because several parameter has been proved to be overestimated by this method. So, the aim of our study is to eliminate the human error done in the analyzing part and also reduce the overestimation caused by the detector. So this is the experimental workflow of the study. First of all, we will use DeepLabCut software to track the animal organ, which is in this case Daphnia heart. And for doing this, we will need a lot video of Daphnia heart. So after we get a lot of Daphnia heart video, we will train the network with ResNet 50, ResNet 101, and ResNet 152. And from that, we will select the best network which is used for further study. And after analyzing part, we will move to the summarizing part. The summarizing part will be done by the help of Microsoft Excel, Python, and VS Code. So in this study we will develop some macros in VBA, which is part of Microsoft Excel, to help with batch processing the output of the DLC program. And the other tool software like Python and VS Code will help us with the calculation of some parameter. After that, we will move to the application, which is for validation of our method by by applying expose the Daphnia with pendimethalin and imidacloprid, which is a pesticide. So to get the heart of Daphnia magna video, we will use the high-speed CCD that is embedded in microscope, and then we will focus on the Daphnia heart here, and we will record it at 200 frames per second. And after analyzing with the program, DeepLabCut will give us the heart wall age here, and it will be tracked by point here. And from this, we can calculate the volume of Daphnia heart, assuming that Daphnia heart have spheroid shape. So we just need the position of long axis here and the short axis, and we can apply the formula for spheroid, which is 1/6 times p times long axis times short axis in the power of 2. And to get the stroke volume, we just need to calculate the volume at end diastolic phase and end systolic phase. And we can also get several other parameters like the cardiac output, shortening fraction, and ejection fraction. And to get the formula for heart rate calculation, we have to get the timing of each beat either at diastolic or systolic volume. And by calculating the average of those beats, we can get the heart rate per minute. And this is the comparison of network performance to track the Daphnia heart by using DeepLabCut. In this case, as I already told before, we use three different kind of network, and it seems that ResNet 142 give the best result, show by the lowest loss rate compared to the other network. And also, ResNet 152 also give us the lowest root mean square error at the end of 500,000 iteration. So in this case, we could say that our model are actually eligible candidate for analyzing the cardiac performance. However, the ResNet 152 is the best one because it shows lowest loss rate and also lowest root mean square error. And next, we compare the DLC performance with previous method, which is the TSA time series analyzer and chemograph method, which is based on ImageJ. And we also compare it with previous mass RCNN method to analyze the Daphnia cardiac performance. And as we can see here, if we compare the DLC method with the mass RCNN method, it will show significant different result, which is in most of the case, mass RCNN method, which is previously done, have given a very overestimate value, even compared to the manual counting method, which is done by ImageJ. However, there's no significant difference observed in heart rate, which is the cardiac rhythm parameter, even given from all the method used. So in this case, although the mass RCNN give overestimation in volumetric based calculation, no significant difference in the timing based calculation, which is the heart rate, the SD1, SD2, and also the heart rate variability parameter. And as we can see here, the LC method give the somehow similar value to either the TSA method or the chemograph method. As we can see here, in diastolic and systolic volume, it gives significant similar result with the chemograph, and in ejection fraction, stroke volume, cardiac output, and also shortening fraction, it gives a similar result with the TSA method. So in this case, we could say that this DLC method gives similar value to manual calculation method. However, as we can see here, DLC is somewhat significantly slower compared to the other method, which is in this case we calculate the uh, time needed for analysis. However, DLC method is equipped with automation to reduce human labor and minimize human error in longer run. So it still can be useful for calculation, for especially for the beginner which has never used the image-based method. And later we will compare the value of cardiac performance parameter after incubation in imidacloprid. And as we can see here, at highest concentration, which is 10 ppm, imidacloprid already alter the stroke volume, ejection fraction, shortening fraction, and even the heart rate of Daphnia magna. However, as we can see, there's no significant change in cardiac output. I think this is because actually the increase in stroke volume is compensated by the decrease in the heart rate, which is a normal phenomenon. And next, this is the comparison for pendimethalin. As we can see here, pendimethalin at even 1 ppm can significantly increase the stroke volume, ejection fraction, and shortening fraction. And it is also followed by some decrease in heart rate, uh, and also it alter the rhythm of cardiac heart show by significantly high SD1 and SD2 value. However, again, we cannot, in this case, we didn't see the significant alteration in cardiac output because even though the stroke volume is bigger, the heart rate is lower, so it somewhat compensate. And next, I will teach you how to use the macro for calculating the cardiac parameter. This macro is divided into three parts, and in each part, in between of this part, we will use the peak analyzer software, which is based on Python, and also the heart rate variability analysis, which in this case we will use the VS Code program. So to use this macro, there's several requirements, a file required, like the formula Excel, which is the inside of the Excel have some formula already embedded in each cell. So the basically the formula, this formula will be copied later to your original file. And also the DLC VBA DLC, which is divided into three parts, and also calculate SD program and which PickFinder program, which is based on the uh, Pythons. First step, as the result name after video analysis consists of long character, we have to rename the file and leave only the important information. And in this case, as you can see here, in my case, I only leave the control name and control the number of animal. And for deleting this, you can either delete it manually or using some rename program like as advanced renamer or something else. The second step is to open the formula files. And actually, in this step, the formula file still doesn't be used. For after you opening a formula file, you need to open a new blank workbook without closing the formula files, like this one. And after that, from the blank workbook windows, open the developer tab here, and then select the Visual Basic. And after that, you will see the Visual Basic windows is pop open, and select Insert, and then choose the module. The fourth step is first you have to open the part 1 of the VBL DLC hard macros. This macro will help you to summarize all the data, the CSV data from DeepLabCut software into one single Excel files. So just copy the whole formula into the windows that open after you open the module in Visual Basic, and then change the the path name of the on this macro. You have to change it to the path of the files that have been analyzed by DeepLabCut. And after you change it, the macro will summarize everything and sort everything based on the file name from the first to the latest one, and it will sort in ascending order. The next step is to calculate the timing of end systolic volume and end diastolic volume using the PickFinder app that has been developed. And in this case, I use Anaconda 3 program, and from Anaconda prompt, I recommend you to activate your environment by selecting the Conda activate and environment name, and open the PickFinder folder using the command cd and space type the file path like here, and then install the requirement to run the PickFinder app by command pip install -r requirement.txt. And after that, to open the PickFinder app, just type python app.py. And this window, it will be open. This is the PickFinder windows. And first of all, what we have to do is to load the Excel. We load the Excel by clicking this and open the file location, which is the location of Excel files uh, that has been saved previously and applied the first part of the macro. And next, after you open the file, the program will automatically calculate the tolerance level for each video, so we don't need to change this part in most of the case. And after that, we tick the exclude on edge box to remove the possible false peak at the start and at the end of video. And then we change everything we apply every setting to the all sheets, or actually we can select just one sheet, but since we in this case we want to do the batch calculation, we apply everything to the all sheets. And after changing then loading the file, just find the pics, click find the pics in, and the pic finder will run the program and find the pics based on the loaded Excel files. And this is the result after we apply the PickFinder function, the PickFinder app. It will tell us the position of end diastolic volume in this case and end systolic volume together with the the volume of the heart at that timing here based on the picoliter per beat. And after that, we open the part 2 of a VBLDLC program, and just copy everything to the Visual Basic application by opening the developer and uh, the developer insert function and open the developer option. And again, just tick the insert and model, and this window will open. The macro will apply the formula from the formula files to calculate the cardiovascular endpoints, as we can see here. And for calculating the heart rate variability, there's some preparation that we have to do. First of all, we have to copy the minimal interval, which is here, which is show the interval of you can either select the minimum interval or maximum interval. It's actually up to you, but in my case, I will select the minimum interval, and then the format should be like this: fish 1, fish 2, fish 3, fish 4, and yeah, just move everything to either ventricle or atrium part. And after that, we run the program by using the Anaconda again, and again activate your environment and open the file, open the app by putting the path of the save app here. And then first of all, we have to install the requirement to calculate the SD1 SD2 by using that program. And after all the program has been done, it will open the calculate SD EPYMB, and just press Control Alt 5 F4 F5 to run every code that has been written here. And it at some time it will ask you the file location of the prepared in the nin step. Just load that file, and then after that, the output folder in the output folder in calculated HD folder will show here, and it will show the SD1 and SD2 at the bottom of the worksheets here for corresponding animals. And actually, this is the optional step, but it will be easier for you to if you done this program. This is the part 3 of DLC VBA. For this program, to run this program, we do the same step as before. Open the developer tools, and then open the VBA option, and from insert, just insert the module. And this program actually used for summarizing. It will get the average and diastolic volume, average and systolic volume, the stroke volume, the heart rate, and cardiac output, and the other parameter into a single summary worksheet here on the corresponding Excel file. So you can either use it or not, but if you want to automatically summarize everything, then it's recommended to use this program. And there's some reminder when you use this program. First of all, never close the formula Excel file until the last step of the formula, and only run the VBA code from the corresponding Excel workbook, which is if you run on the, let's say you run from the formula Excel files, it will give you some error. And it's recommended again to make some Python environment before installing the requirement for the PickFinder and the Calculate HD app, as installing in the base environment will change it and disturb your computer normal process in the future. So the conclusion of this study is ResNet 152 performed better than the other network by showing less loss value and less error rate in both training and testing phase. And one-way analysis showed that the LC method have high correlation with both TSA and chemograph method, which is an image-based method that mostly analyzed done by manual people. And the DSC method is also provided with some macros in VBA to support the process of summarizing the raw data from DLC, and some Python script to enable batch processing and reducing the human error caused by less experienced operator.